Today we will discuss about what is the enzyme and how the enzyme inhibition works. There is three types of the enzyme inhibition: competitive, non-competitive and uncompetitive inhibition. Okay, so let's begin with the class. So what is the enzyme? So as you know that the enzymes are the proteins, right? So it is made up of the amino acid. Like other proteins, it is a kind of also protein, but it helps in the catalysis of something. It is helping in the converting of substrate to product. Okay, like other proteins are like structural protein is there, and some enzymatic proteins say there, some hormones are also proteins. So like that, enzymes are is also a kind of proteins which work as a biological catalyst. Okay, and it accelerate the specific chemical reaction in biological system. It alters the rate of reaction in biological process. Okay, and these enzymes are mainly high molecular weight compounds which is made up of the amino acid chains, and like other proteins it is also made up of the amino acid chain which is linked together via the help of peptide bonds. Okay, and there is a form of the enzyme. Suppose here you can see that this blue color. Okay, when means in the nature there are different type of enzymes is present which require the cofactor for their working. Okay, so that type of enzyme, when means it doesn't bind with the cofactor, then it is called as the apoenzyme. And once it bind with the cofactor of it, this cofactor either can be any kind of protein or any kind of the metal ions. Okay, so when this cofactor bind with this enzyme, then or apoenzyme, then it is called as the holoenzyme. Okay, so this holoenzyme is the apoenzyme together with its cofactor. Okay, and what is the apoenzyme? When means suppose certain kind of enzyme which is inactive, we can say like it is inactivate form of the enzyme, and its activation requires certain kind of the cofactor, then it is called as the apoenzyme. Okay, not all the enzymes are apoenzymes. Okay, that also you have to remember. Like only some enzymes which require the cofactor for their activation, that is called as the apoenzymes. Okay, and once this cofactor is bind to this apoenzyme, then it is make as a holoenzymes. Okay, so this is the like how this enzyme is convert the product into substrate. So firstly, this is the free enzymes. Okay, and there is the two active site of the enzyme. Active site means where the substrate is bind to the enzyme. Okay, so that is called as the active site. Okay, so here two catalytic pocket is there. Okay, and once this sorry, this direction, this is the enzyme. Okay, so this is here the substrate is firstly bind with the enzyme, then it is make the enzyme substrate complex. Once this enzyme substrate complex is make, it creates some kind of transitional phase. Okay, so this in this transitional phase the energy is very much higher. Okay, so means in the free form of enzyme, what happens is, suppose this is the substrate, when it is without enzyme converting into the product, then this transition energy is too high. Okay, so once the enzyme participate in any kind of the chemical reaction, then this what it will do is, after binding with the substrate, it is making the ES complex, enzyme substrate complex, and this enzyme substrate complex stabilize this complex and lower down this transition state energy, and quickly convert the substrate into the product. Okay, so here the next step is the enzyme product complex. Okay, and now the product is bind with the enzyme, so it is required to release the product from the enzyme. So in the next step the product will be released from the enzyme, and it will go to the its target site and perform its function. Okay, so this is how like enzyme help in converting the substrate to product. So firstly, the enzyme is present in the free form, and its substrate is also present as a free form. Okay, and once this substrate is bind to the enzyme, it creates some transitional phase where the energy is very high. Okay, and then this enzyme substrate complex is stabilized, and it convert the substrate into product very quickly after binding with the enzyme. Okay, so in this form, the enzyme the product is bind with the enzyme. So to do their function, the product should be removed from the enzyme. So in the next step, the product will be released from the enzyme, and it will go to its target site and perform its function. Okay, so this is the like how enzymes work. So here mechanism of enzymatic reaction, what I was telling is that here suppose this is the without enzyme, this blue one. Okay, sorry, this red one is the without enzyme. Okay, so here you can see that the transition state energy is too high. Okay, but once the enzyme participate in this chemical reaction, then the substrate to product conversion energy is very much low down. Why it is low down? Because it is bind after binding with the substrate, it is stabilizing the enzyme substrate complex, so that it can easily like down this transition state energy, and quickly convert the substrate to product. Okay, so here with enzyme, the activation energy, this transition state energy is also called as the activation energy. Okay, so activation energy with enzyme, and this activation energy without enzyme. Okay, so here you can see the difference between with enzyme or without enzyme. Okay, so you have to remember like the enzyme can increase the rate of the reaction. Okay, but it decrease the Gibbs free energy or the activation energy. Okay, but the overall free energy here, this is the overall free energy, it will be same. Okay, the only it is decreasing the activation energy. Okay, or the transition state energy, it is decreasing, as well as it is increasing the rate of the reaction, but means at the same time it is not changing the overall free energy. Okay, free energy of the reaction. Here you can see that this is both the line is falling to one another. Okay, so this is the overall energy released during the reaction. Okay, when one substrate is converted into the product, so that time the overall energy or the overall free energy is not changing in the presence of enzyme or in the absence of enzyme. Only the activation, the transition state energy is changing. Okay, and it is because it is low down the transition state energy, that's why the substrate to product conversion is also very fast. Okay, so it can increase the rate of the reaction. So in many exams, like in the statement wise question, it has asked like means you have to give many options, and you have to select like which is the correct option and which is not the correct option. So like that many statement can be given, and from there you have to choose the correct option. Okay, so that thing also you have to remember, like it is only decreasing the activation energy, not changing the overall free energy. Okay, so that you have to remember. So the factors that affect the enzyme activity, so there are several factors that affect the enzyme catalyzed reaction. These are the following. First one is the sorry first one is the substrate concentration. Okay, so suppose you are increasing the substrate concentration. Okay, so like this is the enzyme, and this is only um, three side. Okay, so in the starting, when you give only single substance, then it can quickly convert, because it is having three side for this substrate. Okay, so it can quickly convert this substrate. And with the time when you increase the substrate concentration, now what will happen is at a particular time, with increasing substrate concentration, this reaction will increase, reaction rate will increase, and after certain time it will be in the stationary phase. Why? Because all the substrate binding site of the enzyme is now bind with the substrate. So there is no free space to bind with other means more substrate. Okay, so that's why initially in the starting period, when the enzyme binding site is more present, then with increasing substrate concentration, the reaction rate will be, or the enzymatic activity will increase, and after certain time when the all the binding site will fill with this substrate, then it will go into the plateau phase. Okay, so this that's how the substrate concentration affect the enzymatic activity. Then second one is the enzyme concentration. Okay, so it is the vice versa of the substrate concentration. Like if suppose there is multiple not multiple same type of the substrate but in very large quantity. Okay, so when you give single enzyme to here, then it is only having single site, right? So it will startingly it will slowly means do the enzymatic activity. But when you give the lots of enzyme here, then what will happen? The activity will be fast, right? Because now to accommodate all this substrate, now there is a means enough enzyme is present. Okay, so that's why the activity will be increased. Okay, the another thing is the pH of the media. Okay, so every enzyme require a particular environment to perform its optimal work, right? Because it is a kind of protein. Okay, so every protein is having their pi. Okay, so so pi is nothing but the pH. Okay, so where it is present as a deuteranic form. Okay, so that's why the this protein will work on a optimum pH. Okay, so every enzyme is having the optimum pH where it can function maximum. Like that, temperature can also a initial essential factor for the enzymatic activity. Okay, so it can mean some enzyme can be thermostable, which can work on the higher temperature. Okay, some can be work on the medium temperature, that is the room temperature. Okay, some can be work on the cold temperature only. Like that, the temperature is also affect the enzymatic activity. Okay, and then there is the effect of inhibitors on the enzymatic activity. Okay, so now when you giving the inhibitors, as the name suggests, it will inhibit the enzymatic activity. Okay, so it can be broadly three types. Okay, but here I forgot to mention one more type, that is the competitive, non-competitive and uncompetitive. Okay, so these are the three basic type of the inhibitor which inhibit the enzymatic activity. Okay, in a three different manner. Okay, so these are the factors which affect the enzymatic catalyzed reaction. Okay, first one is the substrate concentration. Like when you increasing the substrate concentration and the enzyme by keeping enzyme concentration constant, then the enzymatic activity will increase for certain period of the time. Okay, and after that, when the all the enzyme active site is bound with the substrate, then it got saturated and it form the plateau kind of graph. Okay, likewise in case of enzyme concentration also, when you keep the substrate concentration constant and increasing the enzyme concentration, then also the enzymatic activity will increase for certain period of the time. Then after that, it will got flattened. Okay, and then there is the effect of pH is also means matters in case of the enzymatic activity. Likewise, the temperature of the temperature of the medium where the reaction is performing, that also matters. Okay, and another thing is the effect of the inhibitors. Okay, so these inhibitor are the three types. First one is the competitive, non-competitive and uncompetitive. Okay, so to identify certain things of the means like enzymatic property, scientist Maclis Manton give one equation, this v naught equal to v max and substrate concentration divided by km plus S. Through that you can identify the maximum velocity of any enzyme, or the any means affinity for any type of substrate. Okay, you can identify certain parameters for the enzyme through this equation. Okay, so this is the Maclais-Menten kinetics, is a model of enzyme kinetics which explain how the rate of enzyme catalyzed reaction depend on the concentration of enzyme and its substrate. Like here we have seen like after means certain period of that time, when you are increasing the substrate concentration, it is increasing, and one particular time of period when all the active site is bound with the substrate, then it is not further increased, it got flattened. Okay, so that is here also saying the concentration of enzyme and its substrate, how it influence the enzyme catalytic reaction, that is explained by the Maclais-Menten kinetics. Okay, and here how it is happening. Firstly, the enzyme plus substrate which is reversibly bind to each other and form the enzyme substrate complex. Okay, this is reversible. Okay, and means this in this in this transition phase, it is very much temporary period. Okay, so it can either go in this direction or it can either go in this direction. Okay, so this ES is formation is reversible phase. Okay, and once this enzyme substrate complex is formed, which then react to irreversibly to form a product and release the enzyme again. Okay, so once it is got converted into the product, it can't be reverse back to form a ES complex. Okay, it is the irreversible process. Once this ES is converted into the product, it can't be go back to the ES complex. Okay, but in case of ES complex, is it can dissociate from each other, and it can form the free enzyme as well as the free substrate. Okay, so this is how the enzymatic reaction is work. Okay, so there is certain terminology that you should know. Like first one is the Vmax. Okay, so what is the Vmax? This is the maximum rate of the reaction. Okay, when all the enzyme active site are saturated with the substrate. Okay, so this is the Vmax. Okay, here in the graphs, here you can see this is the Vmax. Means this is the higher velocity one enzyme can achieve. Okay, with a particular type of the substrate, this is the highest velocity that can achieve by enzyme. Okay, so that is called as the Vmax. Okay, and the second thing is the Km, which is more important. Okay, so it is also called as the Maclais constant. Here you can see this is the Km, this is the Maclais constant. Okay, so what is the mean of Maclais constant? It is the substrate concentration, because it is falling in the x-axis. Okay, so in the x-axis there is the substrate concentration, and in the y-axis there is a reaction rate. Okay, so this is the reaction rate. So Km is the substrate concentration at which it is the measure of the affinity and affinity for enzyme for its substrate. As lower the Km, the more efficient the enzyme is at carrying out its function at a lower substrate concentration. Okay, so it is the half of the substrate it is the substrate concentration where one enzyme can achieve its half of the maximum velocity. Okay, in other word, what we can say is Km is nothing but it is showing the affinity of a particular enzyme. Okay, for a particular kind of substrate. Okay, if suppose this Km is low. Okay, suppose this Km is low, suppose this is 1. Okay, and this is 2. Here in the right direction it is increasing, suppose. Okay, so if suppose Km is 1 or Km is low, it means at a lower concentration an enzyme can bind perfectly with the substrate. Okay, so it means it is suggesting that the enzyme is having higher affinity for that particular kind of substrate. That's why it is binding at a very low concentration of the substrate. Okay, if the Km is high, it means the binding affinity with the between the enzyme and substrate is not too good. Okay, binding affinity means the bond which is forming between these two enzyme and substrate. Okay, so if the binding affinity is high, it means the Km value should be low. Then only means at a lower substrate concentration the enzyme is bind perfectly with the substrate. Okay, so the substrate concentration at which the reaction rate is 50% of the Vmax. Okay, so it is the substrate concentration where the enzyme reaches its half of the maximum velocity. Okay, and this is the Maclaurin-Maintain equation. V initial velocity equal to maximum velocity into substrate concentration divided by, this is the Km. Okay, Maclaurin constant plus substrate concentration. Okay, so when you solve this equation, you will get the initial velocity of the any kind of the enzyme. Okay, or one of the thing you can identify, like what is the substrate concentration, what is the Km, what is the Vmax. Okay, through that formula you can identify. Then the next terminology is the kcat. Okay, so this kcat is the catalytic constant or the turnover number. Okay, so it how means what it is describing is like how quickly an enzyme can act. Okay, so means if suppose 1 unit time, how means one enzyme can convert how much substrate into the product. Okay, so means that is showing like how quickly one enzyme can works. Okay, so that is the kcat or the catalytic constant. Okay, and that is how fast the ES complex proceeded to the E plus P. It represents the number of substrate molecule transferred to the product in a given unit of the time by a single enzyme molecule. Okay, so means it is in general term, if I will say like how enzyme means how much quickly it can convert the substrate to product. Okay, so means it is indirectly defining the time required to converting the substrate to product. Okay, so that is the kcat. Okay, so this kcat is we can find out by the Vmax divided by and total enzyme concentration. Total enzyme concentration in the sense when the enzyme is completely bind with the substrate. Okay, at its active site. Okay, so that is called as the total enzyme concentration. So through that you can identify the kcat or like how quickly it can convert the substrate to product. Okay, the next thing is the kcat by Km. Okay, so this kcat by Km is called as the catalytic efficiency. Okay, means it tells you how rapidly the enzyme binds and how quickly it turn over. Okay, both the thing it is defining, and these two things defines the catalytic efficiency of the enzyme. Okay, here kcat is defining like how quickly one enzyme can works, and Km is defining the binding affinity with that particular kind of substrate. Okay, so in this kcat by Km, okay, divided by Km, what it means, kcat should be high. Okay, and this Km should be low. Then the overall this fraction will give you the larger value, which will suggest the catalytic efficiency of this enzyme is high. Okay, so Km should be low. Okay, because as we know that low Km means higher binding affinity. Okay, and higher catalytic constant means it is quickly converting the substrate to product. Okay, so this is the catalytic efficiency. Okay, and this is the Maclaurin-Maintain graph, where you can see that with increasing substrate concentration, it is initially it is increasing, and after certain time when you are increasing the substrate concentration, it is not further increasing the velocity because all the enzyme is saturated with the substrate already. Okay, so that's why the reaction rate is not increasing. Okay, now the inhibition part. First one is the competitive inhibition. So in case of competitive inhibition, competitive means what? Means when two things are competing for the same site for the binding. Okay, suppose this is the substrate. Okay, here in this diagram if you will see, this is the only single place is there. Okay, in the enzyme there is the single place, and substrate as well as the inhibitor both are competing for this site for binding. Okay, so that is called as the competitive inhibition. Okay, so when they both are competing for this particular site, it means they both should be homolog to each other or the analog to each other. Okay, means they should be structurally similar, then also then only it can accommodate this particular place, right? Means we know that every enzyme is like lock and key concept. Every enzyme is having a specific site for the substrate, right? So if inhibitor should be if inhibitor is competitive, it means it should be accommodate in this active site of the enzyme, and for accommodate in this active site of the enzyme, it should be resemble the substrate of this particular enzyme, right? So that's why the competitive inhibitor is a substance that compete with the substrate for the same active site, and this inhibitor are substrate analog, okay, that bind to the substrate binding site of the enzyme, or which we called as the active site. Okay, so competition occur between the inhibitor and substrate for the binding to the enzyme. Okay, and in this case what will happen is the kinetic of the reaction will be remain same, and the km is increases. Okay, means what it means, the Vmax is remain same and the km is increases, because here there is a single site for binding, right? So in some of the enzyme the substrate will bind, and some of the enzyme the inhibitor will bind. Okay, so like if we see in case of the means there is two probability, either substrate can bind or either inhibitor can bind, right? So to bind with means the probability for binding on this two thing is 50-50, right? So if suppose km is increases, means the binding affinity will be low, right? Binding affinity for substrate, because here the another competitor of this substrate is the inhibitor. It is also having the same capability to bind with this active site. Okay, so that's why the enzyme affinity for binding to this substrate is getting lower, because now it is its affinity is divided. It can 50% bind with this inhibitor and 50% bind with this substrate. Okay, so that's why its affinity is decreased. Affinity is decreased means km is increased, right? So that's why in this case the km is increases. In case of the competitive inhibition, the km is increased, but the Vmax remain constant, because whatever the substrate will bind to this enzyme, that will automatically get converted into the product. Okay, so the velocity or the reaction rate will not change, only the affinity for this enzyme will be changing. Okay, because here the inhibitor is present, so there is equal chance to bind with this active site. That's why it is increases the km value and decreases the binding affinity. Okay, and this type of inhibition can be overcome by increasing the concentration of substrate. Okay, so increasing concentration of substrate means what, like if suppose there is a more substrate, okay, and less inhibitor, then binding with this particular site is more, means the probability for binding to this site will be higher for what, for substrate only, because its concentration is high, right? So in the medium, if substrate concentration is higher, then it will obviously more competitive than the inhibitor to bind with this active site. So that's why it is a reversible binding, so which can be overcome by the increasing of concentration of substrate. Okay, so that is called as the competitive inhibition. There is several example of the competitive inhibition. Most common example is the treatment of methanol poisoning. Okay, so what is methanol poisoning? So methanol and ethanol both is the substrate for the alcohol dehydrogenase. Okay, so when this methanol go inside our body through alcohol dehydrogenase, it get converted into the formaldehyde, which is toxic for our body. Okay, so to overcome this situation, what generally people do is they give the higher dose of the ethanol. Okay, so what will happen if the ethanol concentration will be higher inside our body, then definitely it will be more competitive in comparison to the methanol. Okay, so ethanol go and bind with this alcohol dehydrogenase, and it is converted into the acetaldehyde, which is non-toxic to our body. Okay, so in that way we control the methanol poisoning. And means these examples also you have to remember, like many exam point of view it has been asked, like what this type methanol poisoning suggested which type of inhibition, like that it can be asked. So every example also you have to look. Okay, so this is the competitive inhibition, where the enzyme and substrate the substrate and inhibitor is competing for the same site of the enzyme. Okay, and once the substrate concentration is higher, then the means binding, the probability of binding to the enzyme is higher for the substrate in comparison to the inhibitor. Okay, so that's why the inhibitor, this type of inhibition can be overcome by increasing the concentration of the substrate. Okay, so this is the competitive inhibition. Then non-competitive inhibition. So in case of the non-competitive inhibition, what is happening, there is no competition between the substrate and inhibitor, because the inhibitor bind to the enzyme other than the substrate binding site. Okay, so there is a kind of allosteric site which is called as. Okay, so this allosteric side now go the inhibitor and bind. And one more thing, this inhibitor, non competitive inhibitor, can bind either to the free enzyme or the enzyme substrate complex. Okay, it can bind to both ways. It can bind to directly the free enzyme, it can bind to in the enzyme substrate complex also. Okay, so what it is doing is, in case of the non-competitive inhibition, so that's why it doesn't require here that the inhibitor should be homolog or analog to the substrate, because it is binding to at the different site of the enzyme. Okay, and that's why here the Km will not change, because here the binding site is different for both the thing. They are not competing to each other for binding, right? So that's why the Km will be constant here. But once this inhibitor bind to this enzyme, then what happens is it changes in the three-dimensional structure of this enzyme, so that it can't convert the substrate to product. Okay, so in the normal case it is converting the substrate to product, but when the inhibitor bind to the enzyme, then it can't convert it the substrate to product. Okay, so that's why the Vmax, the velocity or the reaction rate will be decreased in case of the non competitive inhibition. Okay, but the Km remains same, because here the binding site is different, the substrate binding site and the inhibitor binding site is different, so that's why they are not competing to each other. So that's why Km remains same. Okay, but it is inhibiting the means conversion of substrate to product. That's why the reaction rate will be changed. Okay, it is decreased. Okay, and here binding of substrate and inhibitor are equal, because here they both are bind to different site. Okay, and the inhibitor changes the conformation of enzyme after binding, so that substrate can't bind to the enzyme. This is one possibility. Suppose in case of free means enzyme bind with the inhibitor, then it can't bind to the substrate, and once this in case of enzyme substrate complex, if the inhibitor bind, it can't change the it can't make happen the conversion of substrate to product. Okay, so there is two way of the non-competitive inhibition. Okay, so these are the example for the non-competitive inhibition, where the deoxycycline is a non-competitive inhibitor of proteinase enzyme of bacteria. Okay, so it in this deoxycycline going and bind to the proteinase enzyme allosteric site, and means hamper the conversion of substrate to product. Okay, like that heavy metal poisoning. In case of the heavy metal poisoning, there is that SH group containing enzyme at allosteric site will be distorted. Okay, because here conformational changes is happening, so that's why if SH group will be not there, then the substrate won't be bind to the enzyme. Okay, then it can't be converted the substrate to product. So this is the non-competitive inhibitor. Okay, so the third one is the uncompetitive inhibition. So what is the uncompetitive inhibitor? So this is the type of inhibition which is seen in the multi-substrate reaction. Okay, and it is very rare type of the inhibition. Generally, most commonly is the competitive and non-competitive. Uncompetitive very rarely occur. Okay, and unlike the non-competitive inhibitor, in case of the uncompetitive inhibitor, what is happening is inhibitor only bind with the enzyme substrate complex. Okay, in case of the non-competitive inhibitor, what is happening? Inhibitor can bind with the free enzyme as well as the enzyme substrate complex. But in case of the uncompetitive inhibitor, the inhibitor only bind with the enzyme substrate complex. Okay, and once it is bind with the enzyme substrate complex, it won't allow the enzyme to convert substrate to product. Okay, in this case, the Km as well as the Vmax both will be decreased. Okay, so means why it is decreased is Km is decreased means it is increasing the affinity. Okay, binding affinity it will increase, because here Km is decreasing. Why? Because once this inhibitor bind with this enzyme, it is stabilizing this substrate and enzyme complex. Okay, and so it is increases it increases the binding affinity. Okay, so it is work as a like cooperative manner. Okay, but it won't allow the enzyme to convert the substrate to product, so that's why the Vmax is also decreases. Okay, Km is decreases because it is increasing the stability with the substrate, so that's why Km is decreases, and binding affinity for the substrate is also increases. But the Vmax, because it is not allowing the substrate to convert into product, that's why the Vmax is decreased. Okay, so the example of this question is this inhibition is inhibition of the lactate dehydrogenase by oxalate. Okay, and inhibition of the alkaline phosphatase by the L-phenylalanine. Okay, so this is the two example of the uncompetitive inhibitor. So this is the rare type of inhibition. What in case of the uncompetitive inhibition, what is happening? The substrate is the inhibitor is bind with only enzyme substrate complex, not the free enzyme. Okay, like in case of the non-competitive, where the inhibitor can bind to the free enzyme, here it can't. Okay, here only bind with the enzyme substrate complex, and once it is bind with the enzyme substrate complex, it increases the stability, or it increases the binding affinity of this enzyme for this substrate. Okay, so that's why the Km will be decreased, and but it is not allowing this substrate to convert into product, that's why the Vmax will be decreased. Okay, so this is the Lineweaver plot for the enzyme inhibition. Here you can see the in case of the competitive inhibition, the Km here it is the Km this is the with presence of inhibitor, this red one, and this blue one is the no inhibitor. Okay, so this is the normal one. Okay, so it is in the y-axis there is velocity. Okay, so that's why both the line is crossing on the same dot. Okay, on the y-axis, that's why the Vmax is unaffected. Okay, but here in case of the Km in the x-axis, when you extrapolate these lines, you can see that with the presence of inhibitor, the Km is decreased. Okay, so this is the Km is decreased. Okay, because it is present as a inverse form, 1 by S. It means the right side will be lower value, left side will be higher value. Okay, so that's why the inhibitor will be in the presence of inhibitor, the substrate means the Km will value will be sorry, I wrongly said, like km is increased, right? So in the right side there is increased value, and in the left side there is decreased value, so that's why the km is increased in case of the inhibitor. Okay. And in case of uncompetitive inhibition, both the graph is parallel to each other, here you can see. Okay, so it means the vmax is decreased as well as the km is also decreased. Okay. But in case of the non-competitive inhibition, what is we are seeing is the km is constant, because they are meeting in the same point. Okay, so the km is constant and the vmax is reduced. Okay, because here 1 by v is there, inverse thing is there, so increasing value will be reducing value. Okay, so that's why this is the lower value. Okay, because this side it is lower and this side it is higher, suppose this is the 0. Okay, so this side it is going, means the value is reducing, more minus term. Okay, this is the more positive term. Okay, so this is the line we were plot for the enzyme, three type of enzyme inhibition. Okay, so related to that, one question has been asked: the catalytic efficiency for an enzyme is defined as kcat, Vmax by kcat, C kcat by km, or kcat by Vmax. What will be the correct answer? Yes, the option C will be correct answer. This is the catalytic efficiency, because kcat is defining the turnover number or the catalytic activity, means how quickly one enzyme can convert the substrate to product, and here kkm is the affinity, binding affinity for any kind of enzyme to a particular substrate. Okay, so these two things is defining the catalytic efficiency of any enzyme. And this kcat is turnover number. This is the wrong means thing, vmax by kcat is nothing. Okay, kcat and vmax is also a wrong equation. Okay, so the correct option will be option C.